Great. We've only got about 20 people. Should I give everyone another minute or so?
The waiting room is empty. Yeah, we could give it a minute just for any latecomers.
Hello, everyone. Welcome to the Grand Round. Just give everyone another minute or so to come in. Okay, good afternoon, everyone, and welcome to the Grand Round. Our Grand Round today is the annual presentation by our medical students. They have a big Inspire conference once a year where a lot of them present some very high quality research. And out of that, we have to somehow try and find the best three. And the best three are all presenting to you today and telling you about what they've been doing. So first off is Bram Lim, who's a fourth year medical student about to start a PhD, who'll be talking about crosstalk between mesothelial cells and group 2 innate lymphoid cells in pancreatic ductal adenocarcinoma. So over to you, Bram.
Thank you, Professor Wilkinson, for the kind introduction, and good afternoon to all. It is a great pleasure to present at the Grand Rounds about a research project which I did last summer at Tim Halim's lab at the CRUK Cambridge Institute. So today I'll be sharing about this project, which is called the crosstalk between mesothelial cells and a specialized type of immune cell known as the group 2 innate lymphoid cells, or ILC2s, in the context of pancreatic ductal adenocarcinoma, the most common type of pancreatic cancer. So among the common adult cancers, pancreatic cancer carries one of the worst survival outcomes. As you look at the panel on the left, the survival has remained virtually unchanged for decades since the 1970s, whereas all other cancers, it has improved. The average survival is less than 6 months from the time of diagnosis, and only around 3% of people live more than 5 years. And one of the reasons for this is that it is usually diagnosed at the late stage when distant metastases has occurred. While immunotherapy has been hailed as a potential treatment for metastatic cancers, such as in melanoma, little is known about the immunology in pancreatic tumors. A recent paper published last year suggested that mesothelial cells in pancreatic tumors can dampen the anti-tumor immune response by recruiting regulatory T cells. Our lab is primarily interested in ILC2s, so we wondered if there were any interactions between mesothelial cells and ILC2s in the context of pancreatic cancer. So mesothelial cells are a specialized type of stromal cells which line the surface of the omentum and visceral organs in the abdomen. And if you look at the middle panel, we see that the omentum is a visceral fold of peritoneum that is composed of white adipose tissue in the middle and enclosed by a layer of mesothelial cells. Anatomically, we know that the omentum is connected to the spleen, the stomach, the pancreas, and the colon. Now, apart from providing a slippery, non-adhesive surface for the movement of visceral organs, these mesothelial cells are increasingly found to play important roles such as in immunology, the recognition of intraperitoneal helminth infection, and they are also capable of differentiating to a wide variety of cells, including fibroblasts, smooth muscle cells, and adipocytes. On the other hand, the ILC2s, they are interesting tissue-resident immune cells, which orchestrate type 2 immune responses at the mucosal and barrier surfaces. They play a key role in protective immunity against helminth infection, in allergy, and in tissue repair. If we look on the panel on the right, we see that the ILC2s respond to cytokines such as IL-25, IL-33, and TSLP. And in turn, they secrete IL-4, IL-5, and IL-13. These cytokines are believed to be involved in the crosstalk between mesophylial cells and ILC2s. So the objective of this project are threefold. First, I investigated the effect of ILC2s on mesophylial cells, which were isolated from the mouse omentum, where I was able to obtain sufficient quantity for primary cell culture. I cultured these mesophylial cells in the presence of ILC2, ILC2 conditioned media, IL-13, and IL-1 beta. And following these treatments, I then measured gene expression changes by qPCR. To appreciate the effect of ILC2 on mesophylial cells in the mouse pancreatic tumors, I performed single-cell RNA sequencing on mouse pancreatic tumors in control mice and in ILC2 knockout mice. And finally, for the third part, I performed immunofluorescence microscopy on mouse pancreatic tumors to determine where these mesophylial cells are found. So, following the mechanical and enzymatic dissociation of the mouse omentum, the mesophylial cells are isolated by flow cytometry. And this involves first removing the CD45 hematopoietic cells, the EpCAM expressing epithelial cells, and the CD31 expressing endothelial cells. So what's left is the bulk stromal population. And from this population, we isolated the mesophylial cells by selecting those which express podopalanin and CD200, but not the stromal marker CD140A, which is also known as PDGFR alpha. So following the isolation of the mesophylial cells, they were immediately plated and ILC2s were added to the culture at day 4. IL-33 was also added to both the experimental and control wells to support ILC2 growth. On day 11, the wells were taken down for qPCR. So what was interesting was that I found that the presence of ILC2 promoted the growth of mesophylial cells, resulting in a higher well confluency, as seen by the growth curves if you look on the top left, and by the InQSite images on the bottom left. Importantly, the presence of ILC2 resulted in reduced expression of ACTOR2, the gene for alpha smooth muscle actin, and increased expression of IL-33 in mesophylial cells. Now, to determine what was causing this change, what we did was we similarly cultured mesophylial cells, but instead added ILC2 conditioned media, also which is labeled ILC2-CM on the figures all the way to the right, IL-13, and IL-1 beta. Notably, ILC2 conditioned media recapitulated the downregulation of ACTOR2 and the upregulation of IL-33 seen in ILC2 co-culture, suggesting that the effect was mediated by something, a cytokine secreted by ILC2s. However, these results also suggest that the effect is not fully reproduced by the canonical ILC2 cytokine, IL-13, and that an additional soluble cytokine from ILC2 might perhaps be involved. To further investigate the roles of ILC2 in pancreatic cancer, single-cell RNA sequencing was performed on pancreatic tumors taken from both control mice and from ILC2 knockout mice. I identified one population of mesothelial cells defined by the expression of podopalanin, CD200, but not PDGFRA. And these are distinct from the two populations of cancer-associated fibroblasts, or CAFs, found in the tumor, namely the actor-expressing myofibroblastic CAFs, also called MyCAFs, and the Ly6C1 expressing inflammatory CAFs, also known as ICAFs. The mesothelial cell population expressed the known mesothelial genes such as LGAS7, WT1, and mesophyllin. What was really interesting is that if we look on the figure, if we look at the violin plots on the right, we see that knocking out ILC2s resulted in reduced expression of IL-33 in the population of mesothelial cells. So just to orientate you to the figure on the right, the red violin plots represent the control mice, and the ILC2 knockout mice are represented by the green plots. And you can see that there is indeed a decrease in expression of IL-33. Now, for the third part of this project, what I wanted to do was determine where in the tissue these mesothelial cells are located, and I used immunofluorescence microscopy to do that. So here we see three different slides: the normal pancreas, the normal omentum, and of course, pancreatic tumors. On the left, in the normal pancreas, you can see that the mesothelial cells form a distinct layer lining the surface of the organ, and they are labeled red and green for protoplanin and algal S7. In the middle panel, we see that there is a layer of mesothelial cells stained red overlying the highly cellular lymphoid clusters stained blue for DAPI. And some of these mesothelial cells appear to be expressing IL-33 in yellow. Finally, in the panel on the right, for pancreatic tumors, the mesothelial cells appear to localize to the tumor margin. So the tumor is sort of labeled by algal S7, as you can see over here. And at the margin, you can see that there is quite a thick layer of alpha SMA expressing mesothelial cells, and they're actually split into two groups. Those labeled in blue, they are the alpha SMA expressing mesothelial cells, and they are found closer to the tumor, so more centrally. And those labeled in white, they are the non-alpha SMA expressing mesothelial cells, which are found more peripherally. So what I think is that these could possibly correspond to the two different phenotypes of mesothelial cells we identified earlier from our experiments. So to summarize, I found that ILC2s enhanced the growth of murine omentum mesothelial cells and promoted the upregulation of IL-33 and the downregulation of alpha SMA. This is likely due to a soluble cytokine secreted by ILC2s that is not the canonical IL-13, so something else. Similar gene expression changes were observed in mesothelial cells, in pancreatic tumors of ILC2 knockout mice, and lastly, the protoplanin expressing mesothelial cells localized to the outer surface of the healthy pancreas and omentum, and importantly to the margin of the pancreatic tumors, separating them from the normal pancreas. So further studies to conduct include optimizing the protocol for isolating mesothelial cells from the murine pancreas for co-culture with ILC2s, and more importantly, to investigate the identity of this ILC2 secreted cytokine that's signaling to the mesothelial cells. An important implication for this study is that blocking the crosstalk between mesothelial cells and ILC2s could potentially switch the immune processes in pancreatic tumors from the pro-tumorigenic type 2 immune response, which appears to protect the tumor, to the anti-tumorigenic inflammatory type 1 response. And all this could be potentially used for the treatment of pancreatic cancers, particularly in the case of metastases. So lastly, I would like to thank Dr. Tim Halim for the opportunity to conduct this summer research project at his lab at the CRUK Cambridge Institute, and Thomas Yip, who is also an MB-PhD student at Tim's lab for his day-to-day guidance. I would like to thank Peterhouse Cambridge, the Association of Clinical Pathologists, and A*STAR Singapore for providing me with funding to carry out this summer project. And finally, thank you to everyone listening today at the Grand Rounds.
Great, thank you very much, Bram. So does anyone have any questions? Please put them in the chat if you do. So I guess I've got a question. You've identified a mechanism that is partly responsible for pancreatic tumor growth.
Mm-hmm.
Is anyone anywhere near thinking of something to actually block that mechanism?
So as far as I know, I'm not too aware if there's any pharmacological treatment targeting these specific cytokines, but possibly one way is you could use small molecule inhibitors to target the receptors or signaling pathways, which have been well characterized. Two is possibly to use antibodies, such as what's currently used, immune checkpoint inhibitors, to block the pathways.
Thank you. I guess somewhere away from that. Any more questions? So one from Tim Rittman. Are there similar mechanisms in other tumours that are known about that could identify potential candidates for the missing cytokine?
Well, that's an interesting question. So our lab actually has previously worked on other tumors as well, such as not really other tumors, but in the case, there was a recent paper just published which showed that ILC2s also play a pro-tumorigenic role in promoting metastases to the lung, and that they changed the immunity from an anti-tumorigenic type 1 to a pro-tumorigenic type 2 immune response, and they appear to switch off the response by NK cells. So perhaps that could elucidate, could give us more insight into how this works.
One from Ken Smith. And this will be the last question for this section. Can you discuss strategies to delete ILC2s if you believe that they're important in tumour progression?
Thank you very much for that question. That's actually a very interesting question. Deleting ILC2s entirely probably might not be a great idea, since they play an important role in the rest of the body, but attenuating its signaling might be something which is more subtle, rather than deleting ILC2s entirely from the patient. Yeah.
Great, thank you. Well, thank you very much, Bram, for a great talk and for answering those questions well. Our second speaker is Ujjwal Kumar, another fourth-year medical student, who is going to talk about SNO-RNAs, a genetic marker to inform the choice of conduit for coronary artery bypass grafting and potential mechanism of action. So over to you, Ujjwal.
Okay, hello, good afternoon, everyone. Thank you, Professor Wilkinson, for the kind introduction. It's an honor to be speaking today, talking about my final year undergraduate research project, looking at SNO-RNAs as a genetic marker to inform choice of conduit for coronary artery bypass surgery, and trying to elucidate a potential mechanism of action for these RNAs. So just a brief introduction to coronary bypass surgery. It's one of the mainstay treatments for treating coronary artery disease and achieving myocardial revascularization, despite improvements in medical therapies as well as PCI, so percutaneous coronary interventions with stents, for example. Despite improvements in these techniques, CABG or the coronary bypass still remains the mainstay of treating coronary artery disease. The indications include persistence of symptoms on maximal medical therapy, where the pattern of coronary disease is not favorable for PCIs. Now, the graph on the bottom right just shows data from National Cardiac Surgery Audit that there's around 15,000 to 20,000 coronary bypass surgeries performed every year within the UK. So elucidating mechanisms for reasons why these don't work as well in some individuals, or trying to kind of optimize patient outcomes is therefore an extremely important field of research, hence its importance. NICE guidelines advocate for CABG surgery in cases where there's multi-vessel disease, for example, with complex anatomies, or specifically when there's left main stem disease. Here, there's shown to be poor prognostic outcomes with PCI therapy, hence the first-line treatment being CABG, as well as CABG is also advocated in place of PCI in certain clinical cases where there are other comorbidities such as diabetes or a low left ventricular ejection fraction. So when performing a coronary bypass, there's a wide variety of different conduits that might be used. So broadly, these can be grouped into arterial conduits or venous conduits. Venous conduits utilizing the long saphenous vein in the leg. They're typically the most commonly used. However, they have their issues, and a lot of these do fail in due course. Now, long-term conduit patency, as I've said, is the single most important determinant of patient outcomes. So if we're able to elucidate why certain grafts fail, we can then choose better grafts and therefore improve long-term patient outcomes. So despite improvements in surgical techniques and pharmacological therapies, saphenous vein grafts' long-term patency is still less than arterial grafts using, for example, the internal mammary artery or the radial artery, for example, just to name a few. So when venous grafts, saphenous venous grafts, are used as conduits, around 3 to 12% of these are occluded even before hospital discharge. Now, these are normally attributed to technical failure intraoperatively or acute thrombosis postoperatively. Between 8 to 25% failed at a year, and between 40 to 50% fail after a decade. Now, these are through a variety of different processes, which I'll then cover in a bit more detail in a minute. However, just to contrast these quite high failure rates of venous grafts, when the internal thoracic artery, the internal mammary artery is used, only 5 to 10% of these fail even after 20 years. They have a much better long-term patency. So there's a few different mechanisms by which grafts may fail. Now, venous grafts specifically have to undergo a process called arterialization, which is the process of sort of vessel adaptation to a high pressure environment that's seen in arterial flow, but not in venous flow. This involves the process of tissue remodeling, which sort of begins quite early on after surgery, and it can lead to future lumen stenosis and atherosclerosis. Now, it's important to note that venous graft atherosclerosis is actually different to native coronary artery disease. It tends to progress and occur much faster, and the disease itself is more diffuse, and the fibrous caps of the plaques are less well defined, or sometimes they're not even present compared to native coronary artery disease. As a result, these plaques are also more likely to rupture, which means that there's an increased risk of future occlusive vascular events, which can lead to distal ischemia and future myocardial infarctions or cerebral infarctions of some sort. And just a little bit on the sort of molecular mechanism of these graft failures. Typically, these occur via activated platelets secreting cytokines and growth factors, which themselves promote smooth muscle proliferation, intimal extracellular matrix deposition, forming this sort of process of neo-intimal hyperplasia. Now, of course, all of these processes I've talked about on a molecular level will require accurate and correct RNA messengers to allow the appropriate synthesis of these signaling factors. And this is where snow-RNAs come in. So snow-RNAs are small nucleolar non-coding RNAs. And typically most snow-RNAs that we know about, they mediate ribose methylation and pseudouridylation of other RNAs, so ribosomal RNAs. So they're involved largely with post-translational modifications via sort of mechanism of sequence complementarity. They're released during pre-mRNA processing under the action of nucleases, and specific snow-RNA clusters, specifically on chromosome 14 in the DLK1-DIO3 region. These clusters are strongly associated with failure of long saphenous vein grafts, specifically a 17 times higher risk. However, these snow-RNAs don't show sequence complementarity to any sites within the human genome. Now, as you remember, I mentioned, sequence complementarity is normally the mechanism that these RNAs work by. So we don't understand and we don't know how these are acting and what's actually causing this association, or whether it's even causative or correlative. So we wanted to investigate that. So our hypothesis was, we know that the spliceosome is a cellular assembly which is involved in post-transcriptional mRNA processing to achieve the final mRNA transcript, which will then be translated to proteins. So our hypothesis was that these snow-RNAs actually hijack or interfere with the spliceosomal function, potentially acting by some means of structural similarity to the spliceosome, since we've already excluded sequential similarity. Now, we know that the U4 RNA is the key regulator of the spliceosomal cycle, and therefore we suggest that this is the key target that we wanted to investigate in this project. So we investigated structural similarity using a variety of different techniques, using computational modeling in 2-dimension and 3-dimension, as well as RNA contact prediction techniques. And we also identified common structural elements between these snow-RNAs of interest and the U4 spliceosomal RNA. So I'm just going to talk you through the figures we've got here. On the left, this structure that is shown here is the 2-dimensional structure of the U4 spliceosomal RNA, and the density of the color of green indicates that a frequency of similarity at that specific site with the snow RNAs of interest. So areas where the color green is deeper, for example, within this region, indicates regions where there are more similar RNAs to that specific site. So using this, we were able to identify specific substructures of the U4 RNA that are most commonly shared with these snow RNAs. Now, interestingly, we found that these common structures and common structural elements tended to fall within structurally and functionally important regions of the U4 RNA, for example, within the active site. And this lends support to our theory that the snow RNAs are interfering with spliceosomal function, since they have extreme amounts of similarity with specific functionally important regions of the U4 RNA. So this led us to look further into sort of duplex formation and seeing how the RNAs might bind to the spliceosome. And we looked at the change in free energy of duplex formation. So the change in free energy is a thermodynamic calculation that allows us to investigate how spontaneous a biochemical process is. So the more negative the change in free energy, the more spontaneous the reaction will be, and therefore more likely to occur under physiological conditions in a cell in the nucleolus. So using this, we were able to rank the duplex formation changes in free energy for all of our 112 snow RNAs of interest. And we were able to rank them in ascending order from the most negative here shown on the left. And these are the top 10 that we identified. And these are the top 4 just showing with the U4 in green and the snow RNAs in red, showing that they are quite capable of forming duplexes along quite long range sites. And they have quite negative changes in free energy. And therefore these reactions will and do occur spontaneously at physiological conditions. So moving on then from 2D structural work, we then moved into 3D structural work, looking at high fidelity 3D structural prediction and modeling. And we were able to do this for our top 20 snow RNAs as we filtered previously by ranking them by change in free energy. We then calculated RMSD values. So RMSD is a root mean square deviation. So what this basically does is it calculates the individual distance between individual atoms and calculates the square root of the mean distances within a specific common element region as previously identified. And by doing this, we were able to rank our 10 candidate snow RNAs again in order of increasing RMSD. So as it seems, the lower RMSD values are therefore more similar because there's a smaller distance between the atoms in these common structural elements, and therefore are the most similar to each other. And these are structurally depicted on the right, with the U4 being shown in blue and the red structures indicating the snow RNAs. Now we can see that these visually, even on simple visual inspection, do have a really similar structure with quite similar 3D conformations. And therefore it's easy to understand how these might be able to imitate each other, or in fact inhibit each other's function in a cell at physiological conditions. So as I've shown, these snow RNAs do in fact show 2D and 3D structural similarity to the spliceosomal snRNA, most specifically and significantly as we've investigated to the U4 spliceosomal RNA. We've also shown that certain sites within the spliceosomal RNA structure of U4 are most frequently shared with the snow RNAs, and these often fall in functionally important locations such as the active site. So this similarity isn't just a sort of artifact, it is actually significant for spliceosomal function. And this similarity means that the snow RNAs might be able to interfere or interact with the spliceosomal function, as well as potentially disrupting the spliceosomal cycle. Now, if we imagine then that we have variation at these snow RNA loci, this might lead to missplicing, as you'll have interference with spliceosomal function, and in fact aberrant spliceosomal function and aberrant splicing, causing pathological transcripts. Additionally, we might also get sort of imitation and increased function of the spliceosome, and therefore we could get excessive pre-mRNA transcript processing via functional mimicry by the snow RNAs. So just to sort of conclude everything, we've said they have a structural similarity, allowing functional mimicry, as well as potential disruption of spliceosomal function and turnover. We know that these snow RNAs are associated with venous conduit failure, but not arterial conduit failure. And therefore this sets up a potential preoperative genetic screening tool that might have a role to inform individual choice of conduit in surgery, such as coronary bypasses, and therefore we might be able to improve long-term patient outcomes by reducing sort of graft failure rates in the long term, as well as therefore reduce the likelihood of re-interventions and recurrence of symptoms, and therefore improving quality of life and ultimately patient survival. So we suggested 2 potential mechanisms for saphenous venous graft conduit failure. So via functional mimicry of U4 by the SNO RNAs, leading to excessive mRNA transcripts, and therefore the process of accelerated arterialization, increasing neointimal hyperplasia, as well as earlier onset and increased rate of conduit atherosclerosis. And lastly, we proposed a mechanism of variation in SNO RNAs causing missplicing, resulting in aberrant RNAs and therefore pathological tissue remodeling following coronary bypass surgery and conduit anastomosis. So thank you very much for listening today. I'd like to thank my supervisors, Dr. Russell Hamilton and Professor Anne Ferguson-Smith of the Department of Genetics for all of their guidance and support, as well as my funding bodies, Pathological Society and Gonville and Keys College, and I'm happy to take any questions.
Great, thank you very much, Joel, for another excellent talk. So does anyone have any questions? And please put these in the chat. I mean, a clear clinical implication is that potentially genetic screening could be used to forecast which is the better type of graft to use. However, surely no matter what your genotype is, you're still better using an arterial graft, aren't you?
There are issues with using arterial grafts, such as the sort of site where the graft is harvested from to start off with. Typically, the sort of leg region has a much greater redundancy in sort of venous drainage, whereas there tend to be some disadvantageous outcomes to, for example, the wrist and the arterial supply of the hand sometimes when the radial artery is taken, for example. So that's often the reason why the venous grafts are still used preferentially by many surgeons, as well as obviously the saphenous vein is much longer. It provides kind of greater flexibility for operative approaches, as well as more options for minimally invasive graft harvesting. So there are still plus points to using venous grafts, but yes, exactly. It remains to be seen whether the advantages of just using arterial grafts as a general policy does outweigh the negatives of it. So that's one of these things that I think there's a lot of guidelines relating, as well as sort of personal preference of surgeons in this case.
Yeah, and I think Chris Powell has asked a pretty similar question about why aren't arterial grafts routinely used in preference to venous grafts? I think you've answered that.
Yeah, I think so.
So actually, what proportion of grafts now are arterial and what proportion are venous?
So it depends on sort of what the indication for surgery is. So for example, if there's multi-vessel disease, they'll typically use one arterial graft and one venous graft. So the most common arterial graft to use is the left internal thoracic artery, given that it sort of attaches already to the aorta at one location. So that's the preferred first artery of choice. But for single vessel disease, the majority, about 2/3 of grafts used for single vessel disease are still venous grafts, owing to easier operative approaches. But once it comes to multi-vessel disease, then typically the majority of grafts used are arterial, with the exception of the single saphenous vein graft used.
Any more questions from anyone? Okay, so if we've got no more questions, we'll move on to our third and final talk.
Great, thank you very much.
Thank you very much. Thank you very much, Joel, for a great talk and answering the questions for us. So we'll now move over to Renuka Chintapalli, who is another fourth-year medical student, who's going to talk about vector-cardiographic tractor-based analysis of ECGs for adaptive real-time arrhythmia detection.
So good afternoon, everyone. Thank you, Dr. Wilkinson, for that introduction. So yes, my name is Renuka, and I'm also a fourth-year medical student at Cambridge. And my project was based on developing a novel vector cardiographic attractor-based tool for adaptive real-time arrhythmia detection. So intermittent arrhythmias occur commonly in a clinical setting, and while not immediately serious themselves, may signal risk for future life-threatening pathology. For instance, atrial fibrillation can ultimately lead to stroke, and for instance, mesenteric ischemia. Therefore, detection of intermittent arrhythmic episodes is desirable for improving prognoses. Existing tools use single lead ECG recordings to reduce dimensionality, but the problem with this approach is that the maximum vector amplitude, which has the most clinical relevance, may be missed if the lead vector is not parallel to the instantaneous cardiac vector at the exact moment in time in which it occurs. And moreover, using only single leads omits much of the information contained within the other leads, including information specific to certain leads and therefore certain arrhythmia types and specific territories, for instance. So vector cardiography, or VCG, provides a solution to this issue. It utilizes 3 orthonormal leads, which are parallel to the 3 orthogonal axes of the body, as shown in the diagram on the right. So Pythagoras' theorem is then applied to calculate the vector magnitude as a 4th scalar lead, and therefore collating that information into a single waveform. This approach therefore optimizes the balance between including important information whilst also reducing the dimensionality associated with analysis of 12 lead data. But in order to be used for continuous monitoring, artifactual baseline variation caused by ambulation, as well as physiological changes in heart rate caused by factors such as respiration, exercise, etc., need to be accounted for. So a method known as attractor analysis may address these issues, which essentially involves representing all the raw data associated with a particular waveform, in this case, a single cardiac cycle, in a bounded space known as the phase space, in which all possible states of the system are represented, with each state corresponding to an individual vector cardiographic signal data point. These measurements are then normalized to the cycle length, thereby removing the temporal dimension so that electrical events can be compared based on their location within cycles rather than based on their absolute timing. As such, the effects of physiological variation in heart rate can be accounted for. Projecting the data to a different dimension then removes baseline variation without distorting the signal's waveform, which can improve accuracy of detection. So in general, this method is useful in the context of making inferences about the behavior of a complex system affected by multiple variables such as the VCG signal, therefore allowing detection and quantification of deviations from the uniform case. So with these thoughts in mind, my aims were firstly to combine vector cardiography, or VCG, and attractor analysis to develop a novel tool for adaptive real-time arrhythmia detection. Secondly, to test the accuracy of this tool. And thirdly, to investigate methods to improve accuracy. So 12 lead ECG recordings were gathered for healthy and arrhythmic patients from the PTV database on PhysioNet, which is an online database, and the St. Petersburg INCART database. The 3 orthonormal VCG leads were then derived from this ECG data by representing the ECG data as a matrix on the left here, and then multiplying by the CUSCRS matrix, which is here, which essentially consists of a table of coefficients representing the weights attributed to each independent ECG lead. The vector magnitude is then calculated using this equation here in accordance with Pythagoras' theorem, as previously mentioned. So the next step was then to generate an attractor, but in order to do this, you need to calculate the delay coordinates from the original trace, each representing a point, a certain fraction called tau of the current cycle length behind any given VCG data point. So as you can see here, tau is usually taken to be a third. Therefore, a particular data point called x in the waveform is related to points y and z, which are 1/3 and then 2/3 of the cycle length away from the current point, which is x here. And in this way, the attractor captures phase relationships within the cardiac cycle. But in order to calculate this tau value and therefore the delay coordinates for each cycle, the current cycle period needs to be extracted by identifying the distance between the R peaks using the Pan-Tompkins algorithm, which applies various filters in order to remove noise and also checks whether the candidate R peaks exceed signal and noise thresholds. And these thresholds are modified adaptively based on recent history. So the cycle periods were then, once found, the cycle periods were divided by 3 in order to find these tau values and calculate Y and Z. So once the X, Y, and Z values were found, the coordinates are plotted in what is known as a 3D phase space, which is seen on the left, and then projected to the 2D space seen on the right. And the effect of this is to essentially remove baseline variation without distorting the waveform. So the original aim of generating these sort of visual representations of the VCG signal as an attractor was to select a method for automated discrimination between normal and arrhythmic cycles in real time, because it's done on a cycle-by-cycle basis, either by applying a template against which the morphology of the cycle is compared, or by doing a point-by-point comparison. But the diversity of attractor morphology for the normal and arrhythmic cycles precluded this method from being used. It also meant that no clear threshold could be decided upon for classification of normal versus arrhythmic cycles based on point-by-point comparisons. So since deviations from statistical regularity in attractor morphology could not be deciphered in this manual method, I decided to use a machine learning approach. This involved using a bidirectional long short-term memory neural network with feature extraction in order to improve classification speed and accuracy. So this network, which is kind of basically represented in this visual here, essentially scans backwards and forwards between the points in a single cycle, thus taking into account the time-dependent nature of the input signal. The features that were extracted are instantaneous frequency and spectral entropy for both the X and Y, or what are also known as the V and W coordinates of the 2D attractor that I just showed. And these both represent time frequency moments pertaining to the VCG trace. So this novel tool is then applied in 2 contexts. Firstly, as a bespoke patient-specific program, which is trained and tested on cycles from a single patient. And secondly, as a generic tool trained on data from multiple patients. So in terms of the results, firstly, I wanted to calculate classification speed. So the mean execution time for classification of each individual beat was 22.41 milliseconds for the bespoke tool, and then 5 milliseconds for the generically trained tool. And the other important factor that was important to look at was classification accuracy. So the bespoke tool achieved a high mean accuracy of about 83%, whereas the accuracy of the generic tool was considerably lower at only about 64%. However, the classification accuracy of the generic tool improved upon addition of more data pertaining to the arrhythmia type on which the tool was being tested. So for instance, the accuracy rose to beyond 70% in traces characterized by atrial fibrillation and Wolff-Parkinson-White syndrome, when more data containing traces of atrial fibrillation and WPW were actually added into the training data. So on the right here is a summary of my novel program for adaptive real-time arrhythmia detection. The proposed system may be executed in real time, owing to detection on a cycle-by-cycle basis with considerably low prediction delays. And this is because the testing times are well within the time window of 300 milliseconds, assuming a maximal heart rate, which means that with each cycle, the classification occurs in real time. The second advantage is that the rapid training times also enable adaptation of the device to new data via on-the-fly reconfiguration of the neural network parameters. This adaptability would allow for compensations for variations in ECG patterns caused by individual environmental and temporal factors, whilst also retaining the capacity to detect different types of abnormality. By contrast, the majority of existing tools rely on manifold higher volumes of training data, potentially making such adaptability less feasible due to the need for specialized GPU hardware to efficiently train the data sets. So the high mean accuracy of the using the model trained on individual patient data of about 83% also suggests that the transformation into VCG maximum amplitude preserves clinically relevant waveform features and deviations, and that the use of attractor analysis prior to extraction of time frequency moments successfully removes vertical deviations, as well as accounting for natural variations in heart rate. Given this high accuracy, the bespoke tool may also be employed for continuous monitoring as part of, for instance, a personal wearable device to assess the frequency of intermittent arrhythmia incidents in a clinical setting, in order to flag areas of abnormality for further analysis by trained clinicians. On the other hand, in a daily setting, it could be used to determine exacerbating factors, for instance, in individual patients, and then apply this data to inform lifestyle modification interventions. In the future, it will be important to verify the testing and training speeds that I've identified here. When incorporated into a simpler hardware device, such as, for instance, a Raspberry Pi system, and then confirm accuracy in a continuous monitoring setting by applying the program to recordings made by something like a Holter device. But overall, two things have become apparent from these analyses. Firstly, that the novel combination of VCG and attractor analysis can be used to learn meaningful relationships in 12-lead ECG data. Secondly, this method therefore has potential applicability for continuous monitoring for arrhythmia in both clinical and day-to-day contexts. Thank you very much for listening. And also thank you to my supervisor, Dr. Hugh Matthews, and the Department of PDN.
Great. Thank you, Renuka, for another great talk. So we have a question from Rudolph Cardinal. Thank you, very interesting. If I have this right, you went from ECG to 2D VCG, then additionally extracted features such as frequency information and maybe context, and fed that plus the VCG into a LSTM classifier. So did 2-channel VCG plus features outperform features or 12-channel ECG plus features?
Okay, that's a very interesting question. So in our comparisons, almost all existing programs that use a similar method to this, as in AI, usually use lead 2 data. There's very few that use 12-channel data, and almost all of them use features as well. So no, it didn't. My program did not outperform those, but I think it's difficult to make direct comparisons between my program and the ones that already exist, because they usually use manifold higher volumes of training data, which incurs the risk of overtraining, which means that the program is kind of designed to specifically to classify the cycles on which it was trained, but not necessarily on a novel piece of data. And also, when they're splitting up the training and testing data, it's often a mixture of cycles from the same patient in both data sets, which means that if the tool was trained on particular cycles, it's likely that it'll see similar cycles in the testing data set, and therefore that'll kind of ramp up the accuracy. So it's difficult to make direct comparisons and say that mine outperformed it, but there's certain aspects of my program that are beneficial in a clinical context in terms of the adaptability, the speed, the relatively low training and testing speeds, and the ability to account for physiological variations in heart rate.
Great, thank you.
Thank you.
Okay, any more questions? And I guess following on from that, I would have thought the 12-lead ECG is absolutely the gold standard. So it would be hard to outperform that, wouldn't it?
Yes, in the sense that it includes all the data that you'd want to have in order to classify a certain arrhythmia type. But the issue is the dimensionality is incredibly high, and that would just cause a machine learning program that's working on the fly to just grind to a halt.
Right.
And
So
also,
too complicated.
yeah, exactly. So and also, if you had 12 leads, you would have to extract features from each of those signals. It would just become extremely slow. So that's the main issue.
Whereas human beings manage a 12-lead ECG perfectly well as FY1s, which does show that computers have got a long way to go.
Yeah, I don't think design programs like this will ever replace clinicians. It's more to flag areas, especially in very long recordings. So 24-hour recordings, for instance,
yeah.
It's to flag areas of abnormality for further analysis.
And do we have any other questions for Renuka? Okay, well, thank you very much for another really good talk and answering that complex question there. And that brings us an end to our 3 presentations. Obviously, if people have got last-minute questions they want to give that they've just thought of, please do put them in. But otherwise, can we please extend a virtual hand clap to our 3 presenters? Again, it's great at this medical school to see how many research opportunities our students get. So thank you for those of you who give them, but then how well they take them up. We're talking among the presenters before everyone else came in today, and it's amazing the amount of publications these students have as 4th-year medical students. Rather embarrassing comparison to myself at the same stage. So thank you very much, students, for doing such a great job in doing the research, presenting it, explaining it clearly. And that's the end of today. So thank you, everybody.